guests, ladies and gentlemen. On behalf of Stellenbosch University Rectorate and the Faculty of Medicine and Health Sciences Management team, I'd like to welcome you to this auspicious occasion, the inaugural lecture of Prof. Soraya Bardien here at Stellenbosch University's Tygerberg campus. We'd like to remind you that this event is being filmed and ask you to please switch off your phones for the duration of the lecture. Now please rise as we observe the entrance of the academic procession. Thank you.
Good evening. Please take your seats. Good evening and welcome. It's a wonderful privilege to welcome you to Professor Soraya Bardien's inaugural lecture tonight. I would like to first extend a special welcome to Professor Bardien's loved ones who were able to join us today. We've got her husband here, Mark, her son, Justin, her niece Nadia, and we are also joined online by her sister Delia and her brother Ridwan. So we are very happy to have you here, and we are also really thankful for your supportive and loving relationship that you had with her to have this remarkable journey and this remarkable career which we're celebrating tonight. Professor Bardien is a leading scholar in human genetics, and she serves as the head of the Parkinson's Disease Research Group at Stellenbosch University. Her research is dedicated to understanding the genetic basis and disease of mechanisms underlying Parkinson's disease. It's a field in which she's made very valuable South African and international contributions. She represents Africa and South Africa on committees of two major international Parkinson's consortia, and make sure that our voices, our African voices, are heard in a global research landscape. She has a PhD in human molecular genetics from UCT, where her academic journey began, and that was with distinction. She was named to the Dean of Science Merit list in both 1987 and 88, and she also received the Max Delbruck Science Prize for excellence as a student. Today, we know that her work has earned some numerous accolades. She was the finalist for the 2021 NSTF South 32 Lifetime Achievement Awards, and in 2018, she received the Distinguished Woman in Science Award. Stellenbosch University also recognized her with several honors, including the Research and Innovation Award in 2024, the Vice Rector's Award for Outstanding Research in both 2015 and 16, and the Rector's Award for General Performance in 2011 and 2014. She's been the recipient of several SAMRC self-initiated research awards for her pioneering work, and this has not only been in Parkinson's, but also in aminoglycoside-induced deafness. She's a key contributor to the SAMRC Stellenbosch University Genomics of Brain Disorders Research Unit and is affiliated with the SAMRC TB Research Unit. So she has made a large scientific contribution. Through her research and groundbreaking work, Professor Bardien continues to shed light on the complex genetic factors driving Parkinson's disease. Her research not only underlines scientific excellence, but has also had a meaningful impact on global health. So please join me tonight to welcome Professor Soraya Bardien to deliver her inaugural lecture.
So good evening, everyone. It's so nice to see all these smiley, happy faces. It's like a family reunion. So, thank you so much for making the time to travel from wherever you've traveled to be here, and the people online who are attending as well to make the time to attend today. I truly appreciate it. So today I'm going to take you through a journey. Before I start with the journey, let me please first acknowledge members of the procession. So I acknowledge Professor Richard Stevens, who's acting DVC of Teaching and Learning, my Dean, Professor Almi Miller, who's Dean of the Faculty of Medicine and Health Sciences. We have Professor Bertie Fielding, who's Dean of Science Faculty, Professor Soraya Sidat, who shares the same name as me, Executive Head of Department of Psychiatry, Professor Jonathan Carr, Head of Division of Psychiatry of Neurology, and Professor Louise Warnig, Emeritus Professor and previous Dean of Science. So as I was saying, thank you for coming out on a cold winter's night to be here. Today I'm going to take you through a journey of how I got here and how some of the trials and tribulations that I went through. And I hope I don't kill you with PowerPoint. So I want to start off with a thank you, though. And thank you says, I'm here today because you've all been there for me. And I really appreciate everything you've done. And I wouldn't be here if it wasn't for all of you contributing in some way. So thank you. So this is the layout of my presentation. Sorry, is it too much light? Should we dim it a bit? Yeah, I think so. Sorry that I had to do that. Okay, so this is the layout of my presentation. There are 4 parts. There's that I've broken it up into. The beginning, that's the part that led up to my PhD. Then what did I do beyond my PhD? Sorry. Then I'll tell you a bit about my Parkinson's disease research. And then part 4 is about me outside of work. Who am I when I'm not here? And then I'm going to end off with some take-home messages. So let's begin. So this is me with my parents. I thought I was quite cute as a child. And I'm the youngest of 6 children. So these are I've got 3 sisters. That's Delia, Shireen, and my sister Shahida. And then, is it projecting? It's still quite dark. Anyway. And then this is my brother Dari and my other brother Ridwan. And so my 3 sisters are quite a bit older than me, so they were like second mothers to me when I was growing up. And my brothers, they played with me and made me gruwelijk. So the journey that I'll tell you in this part starts with school, tells you a bit about university, my work, my travels, my life, and then how that culminated in the PhD. So at school, I was a really bad student, especially in the early years, grade 1 and grade 2. I really struggled with sums. The teacher would do this pattern and mine would go up at an angle. I couldn't do sums, and I remember wetting the tip of my finger and erasing all my wrong answers, which drove my teacher nuts. And if it wasn't for my mother, who sat with me in the afternoons and really helped me to draw patterns and do sums, and then my father later on, because my father, he could do really complex calculations in his head. So he taught me about maths and gave me a love of maths. So if it wasn't for my parents, as well as my brothers and sisters, I may have not gone further than primary school. So then high school, I went to Belgravia High. This is on the Cape Flats. We refer to it as Bally's. Unfortunately, our school lab burnt down. So I actually didn't do any lab experiments at school at all. And then in grade 11, when I was in grade 11, this was during the 1985 anti-apartheid protests, and Belgravia High was actively involved in those protests. I remember one day a Casper drove into our school grounds, flattened our school fence. So for a large part of that year, I actually didn't attend school. And that made grade 12 really difficult because instead of revising work that people do in grade 12, we basically were trying to do catch-up. And here I have to do a shout-out to my biology teacher, Mr. Fadli Wahid, because he was hard on us, But he made me realize that, maybe there's some potential and I can go to university. So I have to acknowledge him for that contribution. So then I applied to UCT, and because I didn't know better, I only applied to UCT for a BSc degree. And luckily I was accepted, otherwise the story might have also ended there. In fact, I've done all my studies at UCT, and I was lucky to get a Cyfritz bursary that covered all my fees because my parents couldn't afford to pay my fees. And as Prof. Miller pointed out, I got prizes. I really, really enjoyed doing a BSc. It's, I didn't have to do history and accounting and all those subjects that I was not great at. And in fact, I got the Max Delbruck Science Prize for BSc students, and I was placed on the Dean of Science merit list. And then I stayed on after my BSc honors degree. After my BSc degree, I did a BSc honors degree in microbiology. This was in 1990. And I was supervised by Professor Doug Rawlings. I'm sure many of you know him. He was an excellent supervisor. And I remember taking him my very empty Petri dishes, and he would whip out his magnifying glass to try and see my colonies. So I did get some success, though, because we were able to write a paper that appeared in Gene in 1991. So then after my BSc honors degree, I had to leave studying. I had to stop, and because I needed to contribute to the household. So I got a permanent position in a diagnostic lab in the Provincial Administration of the Western Cape. And I stayed there for 2 years because it was really boring. I thought, why did I do a BSc and a BSc honors degree to think and learn how to think and then be in a diagnostic lab? So I left that lab and I got a contract position as a research assistant in the Division of Human Genetics at UCT. And that's when I learned the power of genetics. Being able to determine that someone has inherited a disease-causing variant and being able to provide that result to them was amazing. I really, really was hooked. And I thought I would illustrate this with a family tree or pedigree. So in this drawing, I've got dad and mom. So men are depicted by a square, and mom and women by circles. So this mom and dad have 3 children. They've got 2 daughters and a son. And let's say dad has an inherited disorder. So people with a disorder in the pedigree, you have them marked in as you fill in the symbols. So dad has a condition. Let's say it's a cardiac disorder, and he passes it on to his daughter and his son. But he doesn't pass it on to this child. And as I say, it's a cardiac condition, so this son actually dies of the disorder. If you can find out what variant this dad had that he was able to pass on to these 2 children and not that daughter, you can then figure out what he passed on to, what she will pass on to her 3 children. So you can predict which of those 3 children are going to get that disease. And that's, giving people that information allows them to make life-altering decisions. Do they do certain careers if they're going to die young? Should they take out better medical aid or insurance? So when I started working with pedigrees and learning this, I didn't want to do anything else because of the power of this field. So while I was while I was working, I got the opportunity to actually study my MSc part-time. And I did it on a disorder known as retinitis pigmentosa. And retinitis pigmentosa is a disease of the eye. It specifically affects the rods and cones at the back of the eye, and these cells die in people with this disorder. So it leads to severe visual impairment or blindness. It starts when they're young, and by the time they're young adults, they are visually impaired or blind. And my supervisors for this project was Professor Raj Ramasa and Professor Jackie Greenberg. I'm sure many of you have heard of them. And this is the family tree that I worked on. And as you can see, there were lots of people, all the colored in symbols, all the people that had retinitis pigmentosa. Different generations had it, males and females had it. And so we call this autosomal dominant inheritance of retinitis pigmentosa. And I was really fortunate because my supervisors allowed me to undertake a research visit during my MSc studies. And this was to the UK, at the Institute of Ophthalmology in London. It was a 3-month visit. And I was terrified because it was my first ever plane trip. The first time I was going to be alone because I stayed at home with my parents. And I was going for 3 whole months. But it was amazing. I truly enjoyed it. I met so many amazing people. I learned so much about myself as well. And I learned that if you can depend on yourself, then any place can be home. And it was an important life lesson to learn. So during that visit, I did do work. We actually found a new locus or a position for retinitis pigmentosa on chromosome 17q. We wrote up a paper. I actually started writing the paper during my visit. It was published in a pretty prestigious journal called Human Molecular Genetics, and it was only the 8th locus for retinitis pigmentosa worldwide. So I even got an OMM entry that I was very chuffed about. So when I came back, I then upgraded to a PhD degree. And during my PhD, I then refined the locus to a specific region on chromosome 17q22. This is the same chromosome in which the BRCA1 breast cancer locus is, but ours is on a different region lower down on the chromosome. And I published 3 more papers during my PhD where we were able to refine the locus to a 1 centimorgan region, and we were able to exclude a number of candidate genes. So we knew what it wasn't, but we didn't know what it was. And then I undertook 2 more research visits during my PhD. This was to the University of Texas Southwestern Medical Center in Dallas. That was a 3-month visit. And then I also went to Baylor College of Medicine in Houston. That was a 6-week visit. And I have to say sorry to my husband here, and you'll see why on the next slide. So while I was working, I met Mark, an adventurer who even built snowmen. So what's not 1997, and I put the marks there because in the first year of my married life, I undertook both of those research visits. So sorry, Mark. And then during my PhD, I had a baby boy, Justin, in November 1998. And that's Justin, who's sitting in the second row. So yeah, that was a very busy PhD period. And so I was awarded my PhD degree in December 1999, the following year. And I have to boast that not many people have their son wear their PhD gown before them. But on a very serious note, though, I suffered quite bad postpartum depression after giving birth. But during that time, people actually didn't talk about it. Like, no one wanted to know how the mom was doing. Everyone wanted to know how the baby was. So you just sort of bit the bullet and continued, continued working. But on a better note, I'm happy to report that the cause of retinitis pigmentosa in that South African family was actually identified. I wasn't part of the group. This was only done in 2020. And interestingly, there's been a few families around the world that were linked to that position, with more than 300 affected individuals worldwide. And it's actually due to large, complex structural rearrangements on chromosome 17q2. So we wouldn't have been able to identify this in those years because the technology was not available at the time. But I'm happy to say that my work did lead to quite a significant finding years later. So that was part 1. Let's look at beyond the PhD. What did I do? So because I had Justin, we decided to stay in South Africa and not go abroad to do postdocs. But I did do 3 rounds of postdocs in Cape Town. One was at Stellenbosch University, another one was at University of Western Cape, and the third one was I went back to University of Cape Town, where I worked on various disorders. And I put this in red here because just to remind myself that Justin was only 3 months old when I went back to work, and that was quite sad. And I went back full-time to work and put him into a creche. And I wanted to say as well that I think all of these names are pretty well known to you. It was amazing being in their labs, and I learned a lot from all three of them as hosts of my postdoc. But when I was at UCT, I also worked with Professor Bongani Mayosi, and to see his dedication to genetics and also to uplifting people from previously disadvantaged backgrounds is something that always stayed with me. But the other thing I also learned is, or what I would recommend, is that one does postdocs in different places, that you shouldn't stay where you did your PhD because it doesn't look good on your CV, and you learn so much about yourself when you work with different people and different environments. And then while I was a postdoc at UCT, a position became available at Stellenbosch University. And this was in August 2004, where I say enticed, but I'm sure many of you know Professor Valerie Caulfield. She can be quite convincing. She wanted to know if I wanted to come back to Stellenbosch University because they were looking for a human geneticist to start new projects and work with clinicians at the faculty. And this was a 2-year contract. The initiative was known as GenRed. I'm not sure if many of you remember GenRed. Louise does. And that's when I met Professor Louise Bardien, who was always willing to help and was very keen to get genetics, human genetics, back at the faculty. And I'm very proud to say that one of the things I did was start up the BSc Honours in Human Genetics program at the faculty, because there wasn't one when I first started. And to date, we've trained over 100 students who've gone on to do master's and PhD degrees. So I was proud of that achievement. And during that time, I worked with various clinicians as part of GenRed. I worked on cancers, heart diseases, eye diseases, and deafness with various clinicians. And it was quite a productive period. I ended up publishing quite a few papers on these different disorders with these clinicians. And it was fun working with different groups. But then, so after 2 years had ended, I was then appointed as a senior lecturer at the university. I now had a permanent position. And that's when I met the Professor Jonathan Carr, who was working on Parkinson's disease research at the time. And I remembered talking to him about the disease and the way he described it as this terrible disease that affects people when they want to retire, when they're in their 60s. It really intrigued me, and also the way how compassionate he is about his patients really drew me into the research, as well as the fact that working on a disease that affects the human brain and trying to find out why people get this disease, I found very compelling. So during this period, and while I was working on Parkinson's disease, I was promoted to associate professor. That was in 2012. And then I was promoted to full professor in November 2019. And then also during this period, I need to do a shout out to Professor Soraya Sidat and Professor Sean Hemmings. These two are a formidable team. And I've learned a lot from them. And I want to thank them for making me part of their two initiatives, the Imagine group, as well as the Genomics of Brain Disorders unit funded by the SAMRC. So thank you. So let me tell you now the last part, which is my Parkinson's disease research. This is going faster than what I thought. So what is Parkinson's disease? It's a disease in which certain cells in the brain die. These cells are in a part of the brain known as the substantia nigra, which is deep in the midbrain. And this is when they do a postmortem, and you don't have Parkinson's disease, this is what your substantia nigra looks like, and you have healthy neurons. But when you have Parkinson's disease on postmortem, you can see that they've lost those cells. And that's because the cells have died. And the problem with these cells dying is that once they die, they never grow back. So those are the healthy neurons and then dying neurons. So what are the symptoms of Parkinson's disease? Well, the symptoms are slowed movement, I can't read my slide, rigid muscles, tremor, and problems with speech. But in addition to these symptoms, patients also have depression, hallucinations, and apathy and a whole host of other neuropsychiatric problems. So you can really see when someone has all of these movement problems as well as these neuropsychiatric problems, people think that it's due to witchcraft, that this person's been cursed. So in certain, you can imagine in certain communities, there's a lot of stigma about having Parkinson's disease, and people just don't understand it. And aging is a major risk factor, so the older you get, the more at risk you get of developing Parkinson's disease. But it's important to note that Parkinson's disease is not part of normal aging. It is a disease. So what do we know about the prevalence? Unfortunately, it's increasing, and this was a study that did projections of PD prevalence. By the year 2050, we're expecting 25.2 million people around the world to have PD. I actually think this is an underestimate because Parkinson's is also really hard to diagnose, and especially in certain communities and countries, there's not enough neurologists to actually diagnose these cases. And what this study showed is that there's going to be a 112% increase between 2021 and 2050 in the prevalence of Parkinson's disease. And the drivers of this increase is mainly population aging, you know, that worldwide people are growing older, and population growth. So there really is this huge problem, the impending problem of Parkinson's disease, which I don't think enough is being done about. So what are the causes of Parkinson's disease? The aging, I told you, in the aging brain, you're more likely to develop Parkinson's. There's also environmental toxins that cause the disease, and things like head trauma. Then there's a genetic component, and then there's an unknown component. And you can imagine in a disease as complex as Parkinson's disease, there's a lot that we don't know. But our research group specifically looks at the genetics part of the disease because there is a genetic component. And our research is divided up into 3 areas of research. We look at genetic causes. Once we find a genetic cause, we look at disease mechanisms. So how does that particular variant cause the cells to die? What is the disease process? And then thirdly, and this is an exciting area that we've started not so long ago, we look at curcumin as a therapy for rescuing those neurons from dying. So I'll tell you very briefly about each of those parts of our research. So in terms of the genetic causes, we recruit from the neurology clinic at Tygerberg Hospital with Professor Jonathan Carr. We also recruit from private neurologists, such as Professor Rian van Koller. And then I also have to do a shout out here to the research nurse, Debbie Acker, who traveled around the country going to various support groups for Parkinson's disease, recruiting people with the disease. So we recruit across all the provinces in the country, and then we extract DNA from a blood sample that we get from each person. And we currently have a DNA bank for Parkinson's disease of over 2,000 samples. This is people with the disease, their family members, and healthy controls. And we were able to publish the study collection in quite a prestigious journal, Movement Disorders, because we are the Rainbow Nation. And so because of that, our genetics is very interesting, very different to anywhere else around the world. So studying the genetics of any disorder in South Africa is a big bonus. So I thought I'll stop a bit or slow down at this slide because I think it's an important concept to get across. I said we look at the genetic causes, and for most diseases, there are 2 types of genetic causes. On the one hand, you have your causal variants, and on the other hand, you have susceptibility variants. So your causal variants, these are variants that are very rare in the population, but they have a large effect. And that's typically where you'll see families. So because the variant has a large effect, that's why you'll see the person will transmit it to their children and so on and so on. And an example of a variant with large effect would be that the family I showed you earlier with retinitis pigmentosa. And then I said on the other hand, you have these common variants, so they're very common in all of us, but each of them has a very small effect. And so this is typically where you don't see families, but you see isolated cases with this disease, because you have to have several of these small effective variants coming together to be sufficient to give you the disease. An example of this would be type 2 diabetes. So I wanted to point out here, if you see a company offering a test for a disease like type 2 diabetes or essential hypertension or schizophrenia, and they're going to look at 1 or 2 or a handful of variants, please don't believe them. this is not true. So in terms of the causal variants, we've done some work on this over the years with our study participants from South Africa. And we found, unsurprisingly though, that we don't have a very high pickup rate of these variants, either because they don't have a genetic cause, but we have families, so we do think that there is a genetic factor. But it's also probably because, as I said, we, the Rainbow Nation, and the genetics in South Africa is so different that we don't expect to see what everyone else finds, because we typically look here at the variants that were identified in European and Asian populations overseas. So more recently, we started looking at the other end of the spectrum, which is those susceptibility variants. And this is some unpublished work that I'm going to share with you. This work is being done by PhD student Catherine Stepp in collaboration with Nacho Mata from Cleveland Clinic, USA. And in this work, she did a GWAS study to look for these susceptibility variants. So along here is all the chromosomes, and we found a hit on chromosome 14 in this Manhattan, it's shown here in this Manhattan plot. I can't tell you yet what the gene is because the work is still unpublished, but it's on chromosome 14. It's highly expressed in the brain, which makes it a good candidate. And it also seems to be particularly prevalent in African populations and not prevalent in the other populations. So we're hoping that this work gets peer-reviewed and published. So let's move on to the next section, which is disease mechanisms. So as I said, once we find a variant, you can't stop at that place, because now how does that variant cause cells to die in Parkinson's disease? So this is what this part of the work is about. And there are many different theories as to why these cells die in Parkinson's disease. And the theory that I particularly like, my group likes, is the theory about mitochondria playing a big role. So if you look at a typical cell, they have these little structures in them called mitochondria, and mitochondria produce energy. I was in a talk once, and they referred to mitochondria as ESCOM, and I thought that was a really nice analogy. And like ESCOM, mitochondria don't always work well. So sometimes they stop working, they die, and neurons, which require a lot of mitochondria to work properly. You can imagine the brain requires a lot of energy to do its work. When these mitochondria die, the neurons die. And that's the theory we have, that this is why you get Parkinson's disease, because of a mitochondrial defect. So we tested this using study participants from South Africa with Parkinson's disease. We did this taking a bit of skin from the person, growing out their dermal fibroblasts. And then, yes, we did show that. We found that there was evidence for mitochondrial dysfunction in these people with these defects. And we also showed that in other models of Parkinson's disease. So if that is true, if dysfunctional mitochondria leads to the death of neurons, then it makes sense for us to think that if we stopped this process, if you stop the mitochondria from dying, then maybe you can rescue these cells from dying. And I'm sure some of you would know that when mitochondria stop functioning well, they produce a lot of free radicals, such as reactive oxygen species. And that, in turn, it's like a vicious cycle, because the more ROS there is in the mitochondria, the more damages it causes to the mitochondria. So the idea we came up here is, if we use an antioxidant to mop up the free radicals, can we prevent the mitochondria from dying? And I'm not sure if you know that curcumin is a very powerful antioxidant. And we did studies then to test whether curcumin can come and rescue these cells from cell death. So that's the next part, curcumin as a therapy. So I'm not sure if you know, but curcumin is actually a component of turmeric, or bori, what you use in your curries or you use as a colorant. But did you know that curcumin is very easily degraded and very unstable? So when you are drinking your golden lattes, you might be producing golden poo. So we came up with a very clever idea with Professor Atmaya Dubey from UWC to use nanoparticles as a way of packaging the curcumin, before it gets degraded so that we can actually take it to where it needs to be in the body. And nanoparticles are actually used widely for the efficient and safer delivery of therapeutic drugs. And I wanted to also say we do this work with Dr. Liesl Smith, and the two students who work on this project is Amy Buck and Jessica Burns. And this is what we do. We take the curcumin, we package it inside the nanoparticles, and then we add a ligand which will target it to the mitochondria. As I said, it's the mitochondria that we think is the culprit here. And that's how we produce these mitochondrial targeted curcumin nanoparticles. I also wanted to say here, though, that we don't use bori and extract curcumin from that. We actually buy curcumin from as high-grade curcumin from a pharmaceutical company. So then we take this mitochondrial targeted curcumin nanoparticles and see if they can rescue cells from death. This is the work of Amy Buck. So here's a picture of a cell with mitochondria. This is now untreated cells. So this is a diagram of mitochondria, but this is how they actually look in the cell. And then we treat it with a toxin, a neurotoxin called paraquat. Paraquat actually causes Parkinsonism in humans. And you'll see here that the mitochondria change their shape. They start becoming round, and they're ready to burst and die. So the next step we did was we first treated them with the nanoparticles, then we treated them with the neurotoxin, and you can see very nicely that we actually rescued the mitochondria. They didn't end up undergoing this death process. So does this mean that we are ready to apply curcumin nanoparticles to humans? Absolutely not. This work is very promising and tells us we're on the right track, but we have to do these tests in other models and other experiments to show that this potentially is the therapy. But I do think the work is extremely exciting. And thanks to Amy for all her hard work, and Jessica. And then this is another, previously I showed you morphology, but here I'm showing you function. And this is unpublished work as well. And we do this work with Professor Gerald Marman from Medical Physiology, where we look at the function of the mitochondria using an instrument known as the Ouroboros. And we see very similar things. This is untreated cells, and this is routine respiration in the mitochondria. When we treat it with the drug, the neurotoxin, the respiration goes down. When we treat it with our with our curcumin nanoparticle, it rescues it, takes it back to how it was before we treated with the paraquat. So this is very exciting work, and we'll see in future where the experiments takes us. So why do we do all of this work? Is it to publish papers? Is it to get accolades? Absolutely not. We do this for the field of precision medicine. And the way I thought I would explain it is to say that doctors usually make recommendations about disease prevention and treatment based on the response of an average patient. So you all know that you take drugs and it works for you, but it doesn't work for the other person. Sometimes you have really adverse side effects and the other person doesn't. So precision medicine is where we are hoping to take into account people's unique genetic makeup, their environment, and their lifestyle in order to give them a drug that's appropriate for them and the type of disease they have and the pathway that's gone wrong in them. This is already applied quite successfully in cancer, where patients are stratified into subgroups and then they are given treatments that are appropriate for the subgroup of cancer that they have. So that's why we do the work, because in my case, looking at the genetics of Parkinson's disease, it provides this part of the puzzle that we need to be able to provide precision medicine to South Africans. And it's also important that we do the work in South Africa because, as I said, our population is so unique that if we only took the results from other overseas studies, it probably wouldn't be appropriate here. So it's a precision medicine is a way to move away from a one-size-fits-all approach to treating somebody. So as I said before, Parkinson's is not well understood. People think it's part of normal aging. When someone has Parkinson's, they think there's a lot that it's witchcraft. all of those symptoms together is probably because the person was cursed. So we need to do outreach activities to make public aware about the disease and that it is a real disease. So we've made YouTube videos, we've written newspaper articles, been interviewed on the radio, and I also wanted to put in here that we have an exciting collaboration coming up with Professor Karen Wellman, who's the executive head of the Department of Exercise, Sports and Lifestyle Medicine, to raise awareness about Parkinson's disease, because she also looks at Parkinson's and uses exercise as a therapy for PD and as a window into what is happening inside the nervous system. So, the fun part. Who is me outside of work? So anyone who knows me knows I love hiking. That's a picture of me and Marlene. There's Linda. And I've been hiking for a very long time. That's how I met Mark, in fact. And even the scary hikes that Mark takes me on is fun. There's a picture, a day walk that I did not so long ago with Monique and Vuyo. And I want to thank Diane and Ian here for a very exciting 6-day Fish River hike that they allowed me to go with my son, Justin. That was very special to me. Thank you. And then I have 3 cats. The middle one is my working companion. So when I work in the evening, she always sits next to me and snores very loudly. And then I also do gardening, but sadly, I have very little time for gardening. I didn't even prune my roses this year. And the other activity I like doing is spending time with family and friends. And sorry, people, I haven't put everyone's pictures up here, but these are just to represent this, that I do enjoy spending time with my loved ones. So, and I'm getting close to the end here. As you know, this is Women's Month. And it was a coincidence that my inaugural lecture is in Women's Month. So then I thought, I wonder how many female full professors there are at Stellenbosch University. So we have a total of 279 full professors. Of these, 94 are women, only a third, which I thought was quite disappointing. And then my next question was, out of these 94 women, who are from the Black, Colored, Indian, and Asian groups? 23. I really think we can do better here. And I wanted to show the progression scale from a junior lecturer to full professor. I really feel that we should identify women from these groups at junior lecturer and lecturer position and see what they need to help them progress up the ladder. Otherwise, we won't change this in the near future. So here are my take-home messages. The first one is about work-life balance. That's a picture of me working at a picnic. So I wasn't very good at work-life balance. But I think it's really important, especially as you age, to find time to do the things that make you you, not only your work. And then the second point I wanted to make is about female staff with children. As I said, when Justin was only 3 months old, we put him into a creche and I worked full-time, and I regret that now. I wasn't able to attend any of his school activities. I wasn't even able to attend his matric ball activities during the day. I only went to the evening event. And if I could go back, I would do those things differently. As I said earlier, I think you should move after your PhD. There are certain circumstances where you don't want to move. But most people, I think, should move. I think you should publish your work, because someone told me once, if you don't publish your work, you didn't do the work. I think you should embrace reviewers' comments as well, because often on your theses and your papers, often they actually make your work better. Don't see it as, now I have to do this. And I would also recommend that people work within a large research group or team. For most of my academic life, I was mostly alone. I found the funding, I found the students, I supervised the students, I read their theses, and I actually wouldn't recommend that. If I had to do things over, I also think I wouldn't have followed the same path necessarily. And then the next point is apply for awards. We often think we're not good enough, but apply for them anyway. And I've learned so much from each award that I've applied for. And sometimes I'm really surprised when I win them. So I think sometimes we are actually we are our own worst critics. And then my last point is enjoy what you do. Because if you don't, it's time to move on. So here's my acknowledgements. I'm not going to read all of them because you're going to read them faster than me. There was a lot of people to thank. And the funders, the study participants. But if you read through that, there's a group that I haven't mentioned. Those are the postdocs and the students. So thank you to all my past postdocs for all your hard work, your enthusiasm, and your dedication. It was amazing working for you and your contribution towards my work. All the present and past PhD students, the present and past master's students, present and past honors students, and of course the research assistants. So thank you for walking this journey with me. I really appreciated everything you did. So in my last slide, I want to say I stand before you today as a woman from a previously disadvantaged background, as a full professor, despite apartheid. Thank you. No hard questions, Jonathan.
Thank you so much, Soraya. That was beautiful, as I knew it would be. It's been really an honor and a delight to be able to work with you. I'm here just to manage the Q&A. So does anybody have any questions? Can I kick off while people are thinking?
I knew you would.
Let's say I was if I was 25 again, and I came to you and said, I'm thinking about doing genetics. It seemed quite interesting when I did biology at school. What would you tell them? The next generation.
I would say absolutely go for it. I mean, I think genetics really has a role to play in making a difference in how we're going to treat and manage diseases. I really, really think so. So, absolutely.
And do you think any things that sort of stand out for you as where things are going to go?
Yes, actually, this was not a planted question, but so I think I work as I'm a member of a large international consortium that works on the genetics of Parkinson's disease exclusively. And what they do is they recruit from around the world, but their main focus is from underrepresented populations such as in Africa and Asia, South America. And I think it's those consortia that need to come together to make a real difference. I'm not aware of other diseases that has a consortium like that. But I think when you work together and look at all populations and the genetics of all populations together, that's when we'll make real discoveries.
Thank you so much. Does anybody have questions? Yes, please. We're going to bring the mic to you.
You can ask Prof. Carr a question.
No, don't do that.
Pleasure. Precision medicine. Mm.
And we spoke about, you mentioned briefly about how there are actually diseases out there that are actually doing these
Like in cancer.
and certain other areas. What is your opinion, like how close are we getting there, and especially with like diseases like this?
No, so, yeah, that's a very good question.
Could you repeat the question for the online?
So the question is precision medicine that I spoke about. It's obviously, as I said, already being used in certain diseases like cancer, but how close are we for a disease like Parkinson's? Unfortunately, we are a long way off. As I said, we need to look at all these different populations, find the variants in all these populations, and put that all together before we're able to make some good, be able to predict. No, so I think we are a long way off. But what I was trying to say is that if we don't contribute the knowledge of the genetics of Parkinson's disease in South Africa, we are not going to be able to benefit from precision medicine when it becomes available.
Yes, please.
Not a bioinformatics question.
No, it's not. See, I mean, you actually showed that people are getting older,
Yes.
and so prevalence of this. No, and I was struck just by seeing people whom I knew but younger, who I actually had in Parkinson's. And I was just curious, all the kind of studies you work on now, also including people that are younger. It seems like, it's quite harsh to say young person.
So, sorry, just for the online audience, the question is, are we seeing Parkinson's in younger people than before?
So I'm sure this is actually a question for Prof. Carr. I don't know if we're necessarily seeing them in younger people, but obviously you need neurologists to be able to diagnose this disorder, and so if you don't have many of them, you're not going to diagnose them. But within our collection, we do have young onset Parkinson's disease, people as young as at the age of 17 or 21. And then they've got those variants that I spoke about, those rare variants of large effect, because then you see it in the family. So it does happen in young onset. It's typically 5 to 10% of all cases would have the young onset. Am I allowed to ask a question?
Excellent. Thank you. So you spoke about curcumin and turmeric.
Yes.
Do you see in countries, say with high turmeric intake,
Consumption, yes.
Subcontinent of India, for instance, do you see lower incidence of Parkinson's? Was that not known?
Sorry, can I just, so the question was, in countries where there's lots of turmeric in diet, is there less Parkinson's?
That's a very good question. And so as I was saying, because Parkinson's is age-related, and in those countries you typically don't have a lot of people living longer. So I think with time, maybe we'll be able to make that kind of correlation. But right now, incidence is low in those countries. But it might be because they don't live long enough.
Yes, ma'am. Mike's going to come to you.
Thank you so much, Prof. Soraya, for an amazing talk. It was very inspiring. And I often joke and say, one day when I grow up, I'm going to be Soraya. And I mean it. You're really a role model for all of us.
Thank you.
Especially that this was something to So thank you for that. And I was just wondering, what do you consider to be the highlight of your career? And is there anything you've done?
So the highlight part, I really like writing papers. It's my thing. The other highlight, I have to say, is working with my students and my postdocs. For me, it was really rewarding to see how people start and how they end up at the end of their studies. So for me, that was one of the best parts. Would I change anything? I think I wouldn't. I would still work on Parkinson's, but I wouldn't work as the single person. You know, I would have, I think, fought harder to get more people to work with me in a permanent position, because what I've done through the years is people would come and go. Students come and go, research assistants come and go. I've never had another senior person in the lab to work with. So that I would change, yes.
I think we can probably stop on that highlight that sums up everything so beautifully. Thank you again. Could I just ask then for the rectorate response? Thank you.
So a lawyer with matric biology has now the task to try and make sense of this lecture. So I'll try my best, colleagues and friends. Dear Prof. Bardien, Mark and Justin, family, colleagues and friends, an inaugural lecture serves as a platform for the Faculty of Medicine and Health Sciences and Stellenbosch University to acknowledge the esteemed status and expertise of a senior academic colleague. It also provides an opportunity for scholars to showcase their academic trajectory over time. The rarity of sharing a professional milestone with an audience that includes family, friends, students, and colleagues adds to its significance. Furthermore, an inaugural lecture facilitates the dissemination of contemporary topics like Parkinson's disease. Prof. Bardien illuminated a research topic that affects so many people today, as we've heard. I'm sure we all know of one person who had suffered of Parkinson's disease. My late aunt, for example, had Parkinson's. When I prepared for tonight, I was cognizant, whenever I speak, I either reference music or poetry. And I tried to find a person of color, a poet in South Africa, a woman, and I couldn't find any. The best I could find, of course, was Adam Small, but I often then also referenced journeys, and I couldn't find anything. The best that I could find was Anke Kroch, who was very, in a sense, very dark in her writing. Also found some American authors like Audre Lorde, also referenced the apartheid journey in the US. So unfortunately, I had to get to music, and unfortunately, it was either Led Zeppelin or the Beatles. I've already used Led Zeppelin's Kashmir last year at engineering, so I couldn't use Kashmir again, despite the turmeric and the curcumin reference. But just for those who don't know, the song Kashmir has nothing to do with Kashmir between Pakistan and India. It's a metaphorical journey. So I ended up with the Beatles, The Long and Winding Road. And I think it illustrates perfectly the first part of Prof. Bardien's lecture tonight of life's journey, where the journey of life itself with its inevitable challenges. She referenced matric or standard 9 with unrest. I was at that stage in standard 7 at Lukhof in Stellenbosch, and we suffered the same. But her reference to bellies made me chuckle because it reminded me of sports days at Athlone Stadium, doing interschools where people from where us from Lukhof could actually come to the city for a change, as Athlone was seen like the place to be if you come from a rural town like Stellenbosch. So thank you for that. It also, the long and winding road, also, if you look at it in the context of it, there's one line in the song where it says, the long and winding road that leads you to your door. That's something that is very hard to achieve or to get to. And those of us in academia know that every time you think you found the key to that door, the key just simply doesn't fit. And you have to mold a new key to find that door. And I think Prof. Bardien has also perfectly illustrated that. So I think she showed us firstly how unique all of our journeys are, but how so often we can see commonalities in our journeys, and we should focus on that what we have in common when we do our research and our work. Now the difficult part. Now I have to make sense of parts 2 and 3. Genes matter, I think, was the first thing. So I think when she looked at the genetic causes, the distinction between causal and susceptibility genetic variants, and that although the causal genetic variants are fairly minor in the South African population, and then rare in families and so on. She also then took us on a journey of disease mechanisms, and then specifically that mitochondrial dysfunction, i.e. the inability of a cell's mitochondria, or the ESCOM, or the powerhouses of the cell, to produce enough energy for proper cell, tissue, and organ function, is or potentially is implicated in Parkinson's disease. Then to the nice part, food. Curcumin, a principal active compound in turmeric, has antioxidant and anti-inflammatory properties. So here I did some homework. Turmeric is used in traditional medicine in Asian cultures to treat various conditions, but as she indicated, whether there is a link yet or not, further research is required. Then she also mentioned precision medicine, that we have to, or that there's a movement away from a one-size-fits-all approach to treat diseases, that innovative medical approach that uses information about genetic variation, the environment, lifestyle factors to guide healthcare decisions. And then finally, work-life balance is very important. So with such insight, knowledge, and dedication to the discipline, the ground is fertile for further cross-disciplinary research, and I wish her well and her team with that. As academics, I think we have the privilege of what has been termed, quote, the sublime art of investigating facts and occurrences, of discerning truth from falsehood and certainty from doubt, in order for our research to strengthen, straighten, and extend the mind. Prof. Bardien, on behalf of the SU community, I extend my best wishes to you for the practice of the sublime art and the successful continuation of your academic journey. With that,
Colleagues, friends, and family, it brings us to the end of this evening, and a big thank you. Firstly, Soraya, for the wonderful talk and for your journey which you've shared with us. To our guests who have been here from outside, Louise, Bertie, thank you very much for your presence from the science faculty, and Richard, thank you very much for your presence from the rectorate. We really appreciate your presence here tonight. Thank you to all the friends and family who've been here, and thank you to the people who've worked so hard to make this event a success. Megan and her team, Wilma is here. Thank you very much, and I know Jonathan Dale on the Stellenbosch campus also helped with this. We invite you to have some refreshments and a little bit of a celebration with us, and thank you very much for your presence.